Thanks everybody for coming. It's great to see you here. This is it's wonderful to be able to see you all in person. And we have also a virtual meeting going on simultaneously. So I think we have something like 100 guests that are online watching us. So welcome to you all as well. The Salk has really been at the forefront of cancer for decades, and it really began to some extent with Renato Del Becco. And some of you may remember Renato, who was with us for many years, and it started with his work in oncogenes in the 60s. And then after receiving the Nobel Prize and many honors and awards, he was actually the founding director of the Cancer Center and was able to recruit some really spectacular people here to the Institute. And among other accomplishments, he established this leadership forum for cancer at the Salk Institute and was the first to apply for what's called a National Cancer Institute for basic laboratory centers. And there's only 7 of them that have been designated in the country. And we've had ours since 1972, continuously from 1972. I think evidencing the fact that this is a terrific and successful place. The focus really is on prevention, but also diagnostics and treatment. And today, all the speakers that you'll hear are from our Salk Cancer Institute. Many of them are new faculty members that have joined us recently. And the focus of the theme tonight is on metabolism, and this is energy production and those sorts of things that you'll hear a lot about tonight. But the strategies that you'll hear will align with the idea of the collaboration that exists between these amazing scientists in trying to starve, in many ways, the cancer cells into death. And there's a wonderful set of stories surrounding that. I want to right now acknowledge the chair of the Conquering Cancer Initiative and a trustee at the Salk, and that's Tim Schoen right here. And he will be your moderator tonight and will be in charge of the Q&A. So I want to thank Tim very much for his leadership over the last few years. It's been a tremendous help, and I look forward to continue to work with him going forward. It's a very exciting time for science at the Salk. I guess a few weeks ago now, it was at the Symphony that we kicked off the Salk Campaign for the Future. It's a transformative $500 million campaign over the next 5 years to raise funds for the future of the Salk. And we're doing science and planning science in different ways now, and this requires, among other things, new space for expanding faculty, but also for new ways of doing science. And this includes a new 100,000 square foot building in East parking lot. Some of you may have seen some videos of that, or if not, there's actually a wooden replica of it upstairs in one of the rooms up there. And in this 100,000 square foot new building, one of the spots, one of the 4 initiatives that will be funded will be the Cancer Institute. So for the first time, we'll actually have a single home that we can designate as the Cancer Institute. You'll be hearing more from many of us about the campaign going forward, but I just assure you that tonight we'll reassure you that the future is bright for us at the Salk and for our cancer research, and it's an exciting time. I would like at this point to turn the mic over to Ruben Shaw, and he is the director many of you know Ruben, and he is the director of the Salk Cancer Center. So Ruben, can I call you out here? Put my mask on here, get my protocol right. You take yours off.
Okay.
Welcome.
Great. Thank you so much, Rusty, and thanks to all of you who are here with us at the Salk and to those of you who are joining us from Zoom. Tonight, we're in for a real treat. We're going to hear a number of the leading cutting-edge research scientists, professors here at the Salk who are doing work on the front lines of cancer research themselves and in their labs. So the focus of today's talk, as you just heard from Rusty, is going to be on cancer metabolism, or cutting cancer's fuel lines. And we believe that targeting metabolism is a powerful new strategy for attacking and defeating different forms of cancer. And so that's going to be the key focus that you'll hear about today in different vignettes from different people. But to start, I want to introduce the cast of characters who we'll be hearing from tonight. So very first, we have Dr. Sue Keck. Sue is the director of Salk's Nomis Institute for Immunobiology and Microbial Pathogenesis. Sue is a full professor who moved her laboratory to the Salk about 3 years ago now from Yale University, where she had been a professor for a little over a decade. Sue received her bachelor's degree at the University of Washington, Seattle, and then did her PhD at Stanford before going on to Yale. Joining her will be Dr. Sachin Panda, who many of you may recognize, who's one of our esteemed world leaders in circadian biology and the circadian clock. Sachin, as you may know, was born in India, but came to the United States. He did his PhD at the Scripps Research Institute just down the street here, and then did his postdoctoral research across the street from that at the Genomics Institute of Novartis. Our third speaker is going to be Dr. Christian Metallo. Christian is a very recent addition to the Salk. He's a full professor. For the first 10 years of his professor career, he started off as an assistant professor across our side street at the bioengineering department at UCSD. Christian did his undergrad at UPenn, University of Pennsylvania. He went on and did a PhD at the University of Wisconsin at Madison, and finally did postdoctoral work at MIT in the engineering department. So those three are the full professors tonight. Then we have two young star assistant professors who've both started in the past 12 months. So first we have Dan Hollern, who is joining us from UNC Chapel Hill, the University of North Carolina at Chapel Hill's esteemed cancer center, where he did his postdoctoral work. Dan did his PhD work at Michigan State, and like Sue, he is an expert, as you'll hear, in immunobiology and cancer biology. And last but not least, we have the powerhouse known as Christina Towers, who joined our faculty only in the past couple of months. Christy did her undergrad at the University of Miami. She then went and did her PhD at the University of Colorado at Denver, where she also stayed and did her postdoc. And she and her family just moved to San Diego from Denver this summer. So we're very excited that she's here to join us, that Christian and Dan are all here. They've all recently launched their labs or are actively setting up and launching their labs as we speak. So it's going to be a very exciting program for you to hear the latest and greatest as we come up with new strategies to target cancers. So now to tell you a little bit about a little bit of background about the science and a little bit broader about the team and others here at Salk working on this area, we have this video.
I got involved in cancer research after my grandfather was diagnosed with cancer. And I remember the feeling that I had when I learned that there were no treatment options that could help my grandpa. It just devastated me, and it felt so unfair that I knew that science was going to be the only thing that would be able to help people like my grandfather. And so that was what drove me to cancer research, with this idea that we have to develop therapies that provide options to people that have none.
For centuries, doctors and scientists have been trying to defeat cancer, and in spite of intensive effort, cancer has remained one of the most difficult diseases to treat. One of the few things in common across the hundreds of different types of cancer is the fact that regardless of what tissue it started in or which specific genes are altered in it, all cancers start to grow and divide more rapidly than the normal cells around them. And in order to divide and grow so much faster, they have different dietary needs. They have different metabolic needs. They have to take up more sugars, more glucose, more amino acids, and more lipids from our diet or from the neighboring cells. And so scientists now understand that one way or another, tumor cells will rewire the biochemical pathways that make up their metabolism. That's why here at the Salk Cancer Center, we have a collaborative team of experts with different strengths that are attacking cancer from different angles. We are targeting cancer metabolism as a way to attack all cancers, including the hardest to treat cancers.
Every cell in the body has to eat in the same way that we eat food. But a cancer cell starts to become deranged in what it eats and how it grows and how it uses that food. And the goal is to try to cut off that supply line. What we hope to do is just push them over the edge so that they really can't survive.
My lab is trying to understand how tumors can metabolically hijack the immune cells in the environment. For instance, by consuming extra nutrients that the immune cells and the T cells might use, but also by providing ones that they don't need. And so we're trying to figure out how we can reprogram or rewire those immune cells, those T cells, to function better in that environment. The Salk Institute is a hotbed for metabolism research, and coming together with cancer biologists and immunologists and trying to find this intersection of how these cell types are metabolically regulated.
Changes in metabolism can alter how well a cancer patient will respond to therapy. And my lab has discovered that a specific type of immune cell called a B cell delivers the most effective anti-tumor immune responses to therapy. And what makes B cells so special is that they can overcome the many challenges that tumors present, including harsh metabolic microenvironments.
You can't really talk about the role of metabolism in cancer without talking about mitochondria. Mitochondria are structures in your cells that called organelles, which basically take the food that you eat and convert that into a source of chemical energy called ATP that your cells, tissues, and organs utilize to do basically everything they need to do. Mitochondria are also sort of an armory of metabolites that the cell needs to proliferate to survive and to withstand stress. For this reason, cancer cells actually take advantage of this. They co-opt mitochondrial metabolism to allow them to obtain their high growth potential and to survive really the harsh environment of the tumor.
A key part of cellular metabolism is the ability of the cells to recycle some of the nutrients that they've already used up. And this allows them to conserve energy. And just like we think of recycling in our houses, it's a way for the cells to continue to grow with less input and still be able to continuously divide. And you can imagine in a tumor, which lacks a lot of nutrients, it's hard to get oxygen, hard to get blood supply in. Being able to recycle its own nutrients is a huge advantage for the cancer cells. And so being able to target this core metabolic process really may be an insight into how we can kill these cancer cells.
So my lab develops new technologies for studying metabolism. So while a lot of my colleagues are experts in biology and have key questions, we actually develop new tools, mostly using a technique called mass spectrometry. What it does is it really allows us to detect and quantify thousands of metabolites from complex mixtures. And so 20, 30 years ago, these experiments were really not feasible because we didn't have the technology there. Now I can take a diseased cancer cell versus a normal cell, and I can look really broadly across all the metabolites and tell you which ones are changing. Or we can study a specific pathway in greater detail and understand how quickly that pathway is turned on in a cancer cell versus a normal cell.
Having healthy circadian rhythm makes sure that our metabolism, immune system, and cell division are tightly controlled, not too much, not too little. And my lab has made the discovery that we can be master of our circadian rhythm by eating within a consistent window of 8 to 10 hours every single day. And when we do this, our metabolism remains optimum, our cancer cells cannot grow too fast, And that ensures that having good circadian rhythm prevents many types of cancer.
For nearly 40 years, my lab has identified and defined genes called nuclear receptors that control how specific tissues in our body respond to diet, hormones, and exercise. The labs here, while small, the nature of the interactions expands their capacity to do novel work. The nature of the research is so captivating that people here from all around the world just want to work all the time. And that's what makes a difference. It's passion, it's thoughtful approaches, it's instinct. And by putting all those together, you get something really new.
Please welcome Dr. Sachin Panda.
Welcome. Let me begin with a story. When I was a little kid back in India, I grew up with my grandparents. My grandfather was working for Indian Railway. He would work in the evening, late into the night. And my grandmother, being a loving wife, would always give him company by staying awake late into the night, sharing a meal or snack at random time of the day or night. And I loved to play with both my grandparents in the daytime or in the evening. So I thought they were superheroes because they're always ready and always alert, whether it was day or night. But soon after the retirement, within a few years, my grandfather came down with Alzheimer's, and my grandma had liver disease that quickly progressed to liver cancer, and within a few years, they both passed away. And I always wondered, how come my superheroes were so susceptible to this devastating disease? And soon after, I joined the Salk Institute and started working on circadian rhythm, which literally means 24 hours timetable inside our body. Soon after that, I came across this interesting, very shocking report from the World Health Organization, which essentially meant that people who sleep less, eat at random time, and don't have this healthy circadian rhythm are more likely to have cancer. It's actually worse than smoking because smoking makes us prone to lung cancer, whereas breaking our circadian rhythm can make us prone to at least a dozen different types of cancer. So that made me connect how my superhero grandparents were so susceptible to cancer. But at the same time, I became more curious. And Salk Institute is an awesome place because by working with great minds like Ron Evans, Ruben, Alan Sagatilian, I came to appreciate the role of metabolism in cancer. And what I realized is metabolism is actually a three-part story. The part one is what we all know, that when we eat, we use our nutrients to build new molecules and nurture our body, and we call it anabolism. And then there is a second part, that is, a body actually breaks down excess, unnecessary, and toxic molecules, and we call it catabolism. And then the third part is every night a body actually repairs itself so that all the damages are repaired and we wake up fully rejuvenated for a new day. But how does circadian rhythm connect to metabolism? Just like we cannot do everything at once and we have daily timetable, a body actually has a daily timetable of optimum time when these three parts of metabolism should happen. And what happens is when our circadian rhythm is disrupted, then this three-part story gets messy. So our cells may not get enough time to repair and rejuvenate before dividing so that they carry on all the damages, potentially leading to cancer. So then how do we use the knowledge of circadian rhythm for cancer care? And what we're finding is by living optimum circadian rhythm, actually it's in our hand that we can have the master regulator, we can be the master conductor of our rhythms by sleeping enough at night, by eating consistently during the day within certain time period, and then exercise. And when we have healthy circadian clock, we can prevent or reduce the risk of cancer, whereas when we have broken clock, then our risk for many cancer go up. The second thing that we are also discovering is, just like a three-part metabolism happens at different time of the day, a body is able to use the cancer drugs at appropriate time of the day. So by timing various cancer therapies to the right time, we can accelerate cure. And then the third part is, just in the last few years, we discovered that by drugging the circadian clock, or making new drugs that act on circadian clock, we can try to repair the broken clock in tumor cells. But to our surprise, these tumor cells die. For example, glioblastoma and certain kind of breast cancers, those cells die, whereas the healthy cells remain healthy. So now, in summary, what we're finding is, by using the knowledge of circadian rhythm, we can better prevent cancer, accelerate cure, and can also find new drug molecules to cure very difficult to treat cancer, so that everybody can live a long, healthy, and fruitful life. With that, I would like to invite my colleague, Christian Metallo, who will talk to you about how to use the new science of metabolism for new drugs for cancer. Thank you.
Thanks, Sachin. I don't know about you guys, but personally, I think about food all the time. In part because I like to eat, but also because that's what we study in the lab. We try to understand how the food we take in gets broken down into molecules, and we call them metabolites. And we use metabolic tracing to track how these molecules feed our body, provide us with the energy for us to think, feel, and see. But unfortunately, if you have cancer, those molecules will also feed your tumor. So what we try to do in the lab is zoom in on those tumor cells and try to understand what is their metabolic microenvironment like, and how do they use these nutrients to survive and grow. And with some molecular tools, we can peer inside the cell and look at how these metabolites flow and interact. In a lot of ways, these metabolic networks are quite reminiscent of the roads that we drive in every day. We take for granted that we can pull our iPhone out and look at Google Maps to see how long it takes to drive from point A to point B. We want to do this for a cancer cell, to look at how the molecules are flowing so we can cut off those supply lines. The data we use to generate this involves stable isotope tracing, where we mark specific nutrients with isotope tags and then use mass spectrometry to quantify enrichment in different metabolic pools. And from this, we can use the same engineering algorithms used by Google Maps in some ways to figure out what are the most critical pathways. So now I'd like to welcome Christy Towers, who's an expert in one of the most important highways fueling cancer.
Thanks, Christian. So in our lab, we work on a specific metabolic process called autophagy. And we can think of it as just one of the many metabolic roadways that we're looking at here in our zoomed-in cancer cell. So autophagy, which I know is kind of a funny word, but in the Greek, it breaks down to auto, which means self, and phage, which means eat. So it literally means to eat yourself. And it's a cellular recycling process that all cells use to eat or break down their damaged material in order to generate more building blocks that will fuel all of cell metabolism. And we know that cancer cells are especially dependent on autophagy as a recycling process. And we know that because if we block it in cancer cells, a lot of the cancer cells will die. So let's go back to our highway analogy here. And so if all of a sudden we were to throw up a huge roadblock on the I-5, right, it's going to cause a huge traffic jam. It's going to back up the interstate all the way up through North County. Everybody's going to be mad. Everybody's late to work. It's going to affect the entire roadways of San Diego. So similarly, if we block autophagy and we throw up a roadblock here, it doesn't just affect that one pathway, but all of cell metabolism is shut down. And this will cause the cancer cells to die, which is great. The patients are responding, their tumors are shrinking, they're feeling better. But cancer cells are cunning. And what our group has shown is that cancer cells can actually find a way to circumvent this block and to get around it and adapt. And just like if there's a roadblock on the I-5, you pull your phone out and Google Maps reroutes you another way. Well, cancer cells can do that too. And we've found that when we block this pathway in cancer cells, they can come up with a metabolic detour that gets them around the block, reinstates all of metabolism, and now the cancer cells come back. And this, of course, leads to resistance. So now these tumor cells are resistant to the therapy we were treating them with. And this is how patients relapse. Patients that initially were responding to our cancer therapeutics, now they're back in our clinics with relapsed tumors. So what we're doing in my lab is we're trying to use cutting-edge technologies to really understand these intricate networks and how they function together. And so we're using optogenetics, that is using light to manipulate cell signaling. And we're using gene editing tools, which is literally like cut and paste, but for our genes. And we're using single cell tracing, where we can actually map each of these cells and watch them as they adapt and change. And it's with these cutting edge tools that we'll be able to get a really broad understanding of how these pathways are connected, so that we're not just throwing up one roadblock at a time and the cells can detour around it, but instead we can throw up multiple roadblocks, hit all these pathways at once. This will cause the tumors to die in a way that they are not able to come back. They cannot become resistant and they cannot relapse. This is how we will come up with an ultimate cure to cancer. But of course, I've really focused just on what's happening on the tumor cells right now. But I'd love to bring in my colleague and friend, Sue, who's going to tell us that the picture's a little bit more complicated than that.
So you may not have realized, but over the past decade, we have been witnessing two major revolutions in cancer research. The first is in cancer genetics. And by sequencing the DNA of literally hundreds of thousands of tumors, we now know the identity of nearly every mutant gene involved in causing cancer. And that's a ton of information, but believe it or not, that hasn't been sufficient yet to date to lead to curative therapies. And that's in part because of the mechanisms that Chrissy just talked about, the way that these tumor cells are so easy to become resistant to therapies. But the other reason is because cancer cells don't exist in isolation. In fact, what we're showing you here, these masses of just pure tumor cells, this isn't real. This is incorrect. Tumors are actually mixtures of many different types of cells, particularly immune cells that heavily infiltrate these tumors. In fact, the second major revolution in cancer research over the past decade has been in immunology, because we now know that our immune system is a very powerful weapon to attack cancer. And just like our immune cells can sense and destroy and eradicate virally infected cells like SARS-CoV-2, our immune cells can also sense and destroy cancer cells. But obviously that's not what happens all the time. That's why tumors progress, because tumors are extremely immunosuppressive. And not only that, but the tumor cells can actually manipulate and twist our immune cells and co-opt them to help the tumors progress. And so what's really important for you to understand is that the communication between immune cells and tumor cells in tumors can literally determine if a cancer patient lives or dies. And we've uncovered a whole new way of thinking about how immune cells and tumor cells talk to one another within the tumor microenvironment, and what we refer to as metabolic crosstalk. This simply means the sharing and transferring of nutrients between immune cells and tumor cells. But what's important to understand is that the types of nutrients that are present in the tumor can actually dictate if those immune cells are functional and can kill cancer cells, or if they become suppressed and help tumors grow and spread. And so let me give you an example of what I mean by this metabolic crosstalk by introducing you to one of my favorite immune cells, the T cell. Now, there's several different types of T cells in our bodies. This particular type that we're showing you here is a killer T cell. That's why we've labeled it this blood-sucking red color. And these killer T cells are cytotoxic. They can kill virally infected cells. They can kill cancer cells. But to be these serial killers, what these T cells need is the right kinds of foods. They need the right kinds of nutrients. They need good nutrients like sugars or glucose or acetate, which is found in vinegar. And this is what allows these cells to maintain this killer activity. But the problem is that as these T cells enter the tumors, tumors are a very metabolically hostile environment. And the tumor cells can compete for these nutrients. In fact, they can even steal these nutrients away from these T cells, leaving them nutrient deprived and metabolically exhausted. But on top of that, because these tumor cells have become deranged in their own metabolism, they can actually start to produce bad nutrients, nutrients that are harmful to the T cells. And because these T cells are being robbed of the nutrients they need, they start to rely on and consume these nutrients that damage them and inhibit them. And this leads to immunosuppression and the tumors to grow and to multiply. So we're trying to understand the inner workings of these T cells in tumors by understanding their metabolic properties and trying to find new ways to rewire the metabolic activities of these T cells to make them more metabolically fit to persist and to sustain their killer activity in these tumors. In fact, our discovery of this metabolic crosstalk within cancer between immune cells and tumor cells is opening up a whole new area of cancer research that we refer to as cancer immunometabolism. And we're leading this area at Salk. We think this is the frontier of cancer research. And our team has a very unique vision to double down on cancer metabolism because we think this is going to be the promise to decoding the core metabolic features of cancer, the more universal metabolic features of cancer that allow these tumor cells to progress, but also simultaneously evade and suppress our immune system. And so we think this is going to be, by this intersection of working in cancer, metabolism, and immunology, this will be the intersection that can lead to the next generation of therapies. And we hope that you will partner with us on this. And now I want to introduce Dan Hollern, who's another cancer immunologist. As you heard, he's new to the Salk Institute, and he's going to tell us about a whole nother way in which our immune system can fight cancer.
Thanks, Sue. So yeah, this was a great introduction on T cells. And T cells are so important because they're the target of our immunotherapies. Immunotherapies work by increasing the activity of T cells and encouraging them to attack cancer cells. And what my lab has discovered is that we can get T cells and immunotherapies to work a little bit better if we give them some extra help. So let me tell you about a different type of immune cell called the B cell. So B cells are a very important component of our immune system. Just like T cells, they protect us from pathogens like COVID-19, and they also protect us from cancer. And so B cells operate as our body's threat detection system. And when they detect a threat, they use their receptors to find the targets on these threats, pull them in, take them apart, and then bring them over to T cells to determine exactly what that threat level is. And so this is the same process that we use to prepare our bodies to fight against future infection. This is why we get vaccinated, is so our B cells can recognize the virus like COVID-19 in the future and prepare the immune system to go to battle. And this is also a very powerful weapon against cancer. So when B cells find a threat, like cancer, they go and communicate that threat to T cells. And this causes a large expansion of our immune system to go to war against cancer cells. And in fact, when we have patients who have tumors that have B cells and T cells in their cancer together, those are the patients that are most likely to respond to immunotherapy and to live the longest. And this isn't only because they prepare the immune system, but it's also because B cells and T cells work together to direct tumor cell killing. So when a B cell is communicating with a T cell, it's encouraging those T cells to make those killer proteins that Sue just talked about to get the T cells to kill cancer. And in turn, the T cells are instructing B cells on how to make antibodies. And I think that it's really been clear over the last year how important these antibodies can be. Just last year, when the former president of the United States was sick with COVID-19, his life was saved by delivering therapeutic antibodies that could neutralize COVID-19. We're also using antibodies already in our war against cancer. Many of you might know somebody who's been diagnosed with HER2-positive breast cancer. And HER2 is a unique target on the cell surface of cancer cells that can be recognized by therapeutic antibodies that will bind to the cell surface of those cancer cells, recognizing HER2, and cause those cancer cells to die, just as you're seeing here in this illustration. And this works together with T cells. And so there are many more targets present in cancer than just HER2. And that's what my lab is focused on identifying as ways of expanding the targeting of cancer using B cells as a therapeutic device. And one of the key challenges in doing that is we know that there are differences as cancer evades the immune system and spreads to new locations. And so we really need to understand what are those effective targets to go ahead and start killing metastatic disease, which is really what is shortening patient lifespans. And this is a very large challenge and will require a team effort. And so I want to reintroduce Ruben, who has assembled this amazing team to allow us to take on a unique challenge like conquering cancer.
Great. Thank you, Dan, and thanks to all of our panel. As you can see, our panel has a great depth of expertise and very complementary approaches in which they are attacking cancer and harnessing different aspects of cancer metabolism to do so. However, they are not the only people here at Salk who are on the case in this fight. In particular, we have a number of already world-famous gurus in metabolism and in cancer who joined together to give us truly a veritable dream team of researchers that is unrivaled in the United States for this constellation of experts and depth to take on cancer. So we're very excited in this new front in the war on cancer. And at this point, I'm going to turn the controls and start the questions and answer sessions. I'm sure there may be a number of questions and lots of more interesting information to dispense before we go to the reception. So at this point, I'd like to introduce Tim Schoen, who's going to be our moderator. Tim is the chair of our Conquering Cancer Board. He is the CEO of Biomed Realty, and he's also a board member on our board of trustees here at the Salk Institute. So, Tim.
Thanks, Ruben. Have our panel come up. Ruben, Sue, Sachin, Dan, Christy, Christian. Right, got them all. So we got the dream team. Ruben said I could moderate today because I passed enriched biology my sophomore year in high school. He did bring my transcripts in. So, but on behalf of the Conquering Cancer Initiative, which we have Rita and Ellen here tonight and the rest of the committee who's joining us by Zoom, I wanted to ask the dream team some questions. And I did pass Ruben, so I'm uniquely qualified to ask questions. That's as far as I can go. But anyway, we'll get it started. We'll keep it moving. But we know that diet and lifestyle can be big risk factors for cancer. So can we really prevent cancer by changing what we eat? Sachin, I'll get started with you since you talked about the circadian rhythm.
Lifestyle is what, where, and how much we eat, sleep, and exercise every day. And by following optimum lifestyle, and one example I showed in my presentation, we can significantly reduce our risk for cancer. And second, even if one of our loved ones is diagnosed with cancer, combining optimum lifestyle with cancer therapy can accelerate cure, return to normal life. And those who have survived cancer, even following optimum lifestyle, can reduce the risk of cancer relapse. So these are three different ways we can improve cancer cure and cancer quality of life. But at Salk Institute, we're actually drilling down the molecular foundation of optimum lifestyle. For example, we're finding that insufficient sleep or disrupted sleep, just like when I respond to midnight emails from Ruben or Christy, actually disrupts metabolism and confuses the immune system. Similarly, we're just beginning to understand that exercise can optimize metabolism, but at the same time, a muscle can produce new molecules that can protect against cancer. And when it comes to diet, I would actually leave it to our diet guru, Christian.
So obesity is a known risk factor for several cancers, and this is in part because fat is laden with energy. This is why I like to say fat tastes so good.
Yeah, like that brisket that you barbecued the other night. That was incredible.
I've worked on that for longer than I've worked on metabolism. But that energy-laden fat can fuel cancer. So we've been working with Ruben and Alan for years trying to find drugs and dietary regimens that can break this circuit. But as Sue and Dan described, in the last decade, the whole equation has changed, and we need to think about not starving our immune system as well. So in the end, at this stage, like the safest advice is everything in moderation.
Even red wine?
Except tonight.
Yeah, and actually talking about fat, working with Christian and Alan Saglatilian and the mass spectrometry core here at Salk, we've identified that fat is one of the most abundant nutrients in cancer, and our immune cells are actually heavy consumers of this fat. When you go and get your blood test, for example, and you get cholesterol, and you hear about the bad fat and the good fat, we have found that the same bad fat that puts one at risk for heart disease is also found in your tumors, and that can also suppress your T cells. But luckily, we've also found a way that the T cells are importing that bad fat, and if we block that, we can actually help those T cells stay more functional in the environment. So that's an example of a potential new way to think about dietary therapeutic interventions.
Yeah, I think the take home here is that it's really complicated, and it's a complex question, but at Salk, we're equipped to handle these sorts of complex questions. And for example, Christian, Ruben, and I are all working on these recycling pathways, and Dan and Sue are working on these immunology pathways, and we're all looking at the tumor from all these different angles. To be honest, I just don't think that there's another cancer center that has a team like ours to conquer these problems.
All right, Dan and Sue, we'll keep you on the hot seat. You both shared that immunotherapy is revolutionizing cancer care. Some people have amazing responses, and some people don't. Some people respond for a short amount of time, and some don't respond at all. Why don't immunotherapies work every time?
Yeah, that's the golden question of the moment. I think one of the things to consider is, as Christy mentioned, that cancer cells are able to adapt so well. So when they're undergoing changes that allow them to resist, like the autophagy inhibitors that you study, they're also making changes that allow them to avoid detection of the immune system, including reducing their expression of those targets that B cells and T cells would recognize to go ahead and decide to kill cancer. And I know that there's many things that Sue's also looking at that add to this equation. I'm wondering if you could.
Well, yeah, I think, you mentioned one at the beginning where he told us that the people who respond the best to immunotherapy are the ones who already have T and B cells in their tumors. And so the problem we need to figure out, number one, is how can we get everyone to have T and B cells in their tumors? And this is something that we don't fully understand yet. But what we're starting to, in fact, some of the same genes that cause cancer, what we call these cancer drivers, can influence whether or not a tumor can elicit an immune response, or what we refer to as its immunogenicity. And actually, this is where our genetic guru, Dr. Ruben Shaw, he knows a lot about this.
Yeah, this is actually like a cutting-edge question in cancer research right now. So just for example, within different subsets of lung cancer, there's enough clinical data now that patients who come in with one or two specific genes altered in their specific cancer, those patients do not respond at all. Like from the beginning, not even a bit to immunotherapy. And so we're very focused now, we're working with Sue and with others here to identify the specific biochemical pathways in those tumor cells with those specific genes that go wrong, that make them completely resistant from the beginning, and then we want to develop new therapeutics against those targets to be able to reverse and therefore untap the potential of immunotherapy for those subsets of cancer patients. So that's something that we're actually working on, multiple people right now.
Rare mutations. These are pretty
No,
common
no, this
cancer
is like,
patients, so.
yeah, like 30 to 40% of lung cancers right now.
And even my favorite metabolic process, autophagy, has been shown to affect how well immunotherapies work. And in pancreatic cancer, for example, we know that if the pancreatic cancer cells have high levels of autophagy, it helps the tumor cells hide from these killer T cells that Sue was talking about. And I think this is just one of the latest examples that shows that we can't really have this old school thinking of cell biologists that work in this department over here and immunologists that work in this department on a different floor. We have to all work together, and we have to take expert immunologists and expert cell biologists and expert metabolomics and put us all in one room to get all of these collaborations going. I think that's really the only way we're going to make headway, and that's what we're doing here at Salk together.
You know, building on that question, we often hear how chemotherapy suppresses the immune system. So how does it affect the ability of the T cells and the B cells to kill cancer?
Oh, yeah, that's also a big problem. But fortunately, clinical trials are helping us solve that. So it's true that, chemotherapy works so well by targeting rapidly dividing cells like cancer cells. But as I mentioned, when a B cell or a T cell recognizes cancer, it needs to expand its population rapidly as well. And so they would be subject to those same toxicities that allow chemotherapy to work. But what's been a major development is that we can use specific types of chemotherapy in combination with immunotherapies that protect the immune system from those toxic effects. And not only that, what we're learning even more recently is that if we time the delivery of drugs very specifically, we can expand those benefits even further. And what I think that means to me and gives me a lot of hope is that there are people like Sachin who is already working on this problem and can help us decide when is our best time to give immunotherapies.
Yeah, so circadian rhythm has untapped potential for cancer treatment and care. First of all, we are testing this very simple idea that maintaining a healthy circadian rhythm by eating and sleeping at the right time can prepare our body's metabolism in a way that we can tolerate the most powerful immunotherapy and chemotherapy. And the second approach is based on our recent use of data science and machine learning algorithms to find this idea that it's possible that specific cancer drugs may have specific time, whether it's morning, late afternoon, or evening, when they're more potent. So that has put us in a position that we are poised to start new clinical studies in collaboration with other cancer centers to combine healthy circadian lifestyle with the right time of the treatment to reduce adverse side effect and substantially increase the potency of these drugs to cure cancer.
Yeah, go work on that.
Yeah, we're super excited about it.
Okay. We had a question that came in earlier from the audience, and I don't know, I kind of want to stand back from the panel a little bit from this because I'm afraid I might get rushed on this. But if we can make a vaccine in 18 months, why has it taken 100 years to cure cancer?
Okay, well, I'll start this one. But I think, first of all, the entire scientific community is very proud of the decades of work that have laid the groundwork for this very successful vaccine rollout of 2021. But these immunologists and virologists really had the entire pathway worked out, and they understood exactly how viruses like COVID-19 work. And so all they were missing was the exact sequence of this version of the coronavirus, and they could sort of plug and play. So I like to think of it as like a gaming console, right? It's like these virologists had already built this elegant Nintendo system, or I don't know, for anybody out there that's a little younger, maybe an Xbox system, right? So all they had to do was plug in this new sequence and play the game. Well, in cancer biology, we're still building our Nintendo. We're still getting all of these pieces together. And we do have a good understanding. Now we can sequence tumors. but we don't have a complete understanding. And so we're just not quite there yet in cancer biology like we are in virology. And there's just so much more to do still.
Metabolism also contributes to these profound differences. We can all appreciate now that tumor cells and viruses will mutate their genomes to become resistant to therapies. But a virus is much simpler metabolically. It relies on host metabolism to proliferate and expand. A cancer cell comes with it all the metabolic tools that a normal cell typically has. And what we're learning now is that cancer cells can reprogram their metabolism to go into a kind of stealth mode that makes them resistant to chemotherapies and resistant to immunotherapies. And that's what we're really going after. Can we short circuit these processes and break down their defenses?
I think we have one question from the audience, right? Sandy? Okay.
Thank you. Wonderful presentations and discussion. I had a quick question about, or I guess for everyone in the panel. So where do you think the next breakthroughs in cancer treatment will come, and what is Salk doing to get us there?
Great. I guess as captain of the team, I'll start maybe. Sorry, guys. Everybody gets their turn. So I think our team here, collectively, we have a really bold vision for really decoding first principles of tumor cell biology and trying to understand how tumor cells rewire their metabolism that enhances their ability to grow and spread throughout the body, cause metastasis, but also how to evade and control the immune cells. And so we are collectively going to create a map of the metabolic landscape, and we are really moving towards a global view of cancer in that way. If you step back for a minute, as we heard from Sue earlier, the first revolution in cancer care, or cancer therapeutics really, was using technology to be able to decode the sequences of all the genes and then all the genes that go wrong in your tumor so that we could find the specific genes that are mutated in each person's tumor with the hope that we could treat each person's cancer based on the specific genes altered in their cancer. That concept is called personalized medicine or precision medicine. And that's great. That was a game changer. But today, so far, that has only led to treatments for about 20% of cancer patients. Then the second revolution was immunotherapy, which unlocked an unpredictable amount of therapeutic use of the immune system. But still, right now, that is helping another 10 to 20% of cancer patients. So one of the goals of this team in the Salk Cancer Center is really to harness now as a third wave. We truly believe that a deep understanding of cancer cell metabolism is going to provide a third wave of many, tens, hundreds of potential therapeutic options to take out and combine with immunotherapy and targeted therapies to beat back cancer.
Yeah, and that's a really good point, because what you just mentioned, the majority of patients don't respond to immunotherapy or don't respond very long to immunotherapy, but the combination therapies that Ruben just talked to is actually showing great promise. We now have clinical trials and data showing that we're getting upwards of 50, 60, in some cases for certain types of cancer, 70% of people responding for more than 2 to 3 years that had metastatic end-stage cancer, responding to combinations of immunotherapy with other therapies. And so we know that there's great promise in combining these therapies and coming to the mapping this metabolic landscape of cancer that Ruben just referred to. This could be foundational for the field of cancer. And going another level deeper, like what I was talking about, that we really need to understand is how are these immune cells metabolically adapting within these environments? And And how does that get influenced by which type of a gene is mutated in the cancer? So in genotype A, or when you have mutant cancer A, do the immune cells react metabolically in one way, but then in cancer B, do they react in a different way? And that's what we really need to understand to better identify how we're going to combine these therapies and target cancer metabolism.
Yeah, there's so much hope in that idea, because you can imagine making designer B-cell and T-cell therapies that would be off the shelf, meaning any patient could get them, and where you could engineer those B cells and T cells to overcome those challenging metabolic microenvironments, or having receptors to find the targets on the cancers that we need them to, or even to express molecules that will call in very specific types of immune cells to eliminate the cancer. And not only could that help patients live longer, but they could help them avoid some of those harmful side effects that they experience with chemotherapy. So I think this, integration of immunometabolism is really the wave of the future for cancer immunotherapy.
Yeah, I think if we combine powerful technology with a dream team, that opens paths for discoveries, diagnostics, and cure. And we strongly believe that the new wave will come from new technology that will enable us to accurately quantify, detect, and follow tens of thousands of metabolites in healthy tissue and in tumor cells, even down to single cell, at different time of the day or during the course of the treatment. And by doing so, we'll be able to identify new choke points in metabolism. And those choke points will be the target of new drugs or combination of drugs, so that we can effectively and personalized cure cancer. And at Salk, we do have a dream team. More than half of our cancer center members already work on metabolism. So with the right technology and right resources, we're really poised to bring the next revolution in cancer metabolism.
Look, dream team or not, there's not going to be any magic diet that cures everyone's cancer. But the hope is, with these comprehensive data sets, if we can build a map of tumor metabolism, of immune cell metabolism, this will allow clinicians and patients to navigate to the most effective therapies. This is going to be a complex problem, but I think it's one we can solve by getting a systems level understanding of the tumor microenvironment, which is what we're going to do.
And I think trying to go after this systems level and really trying to get at these metabolic pathways, we have to have the technology to do that. And we're realizing the problem's way too big to be looking at the bulk tumor anymore. We really need to be able to dive deeper and look at each cell, so we can look at these different metabolic networks. How are they interacting with each other? How are the immune cells interacting with the tumor cells? But to do that, we need technology, and Salk is investing in that type of technology. And just now, we really want to buy this new cutting-edge live cell imaging device that will allow us to peer into the cell at a single cell level, but also at high throughput, so that we can look at thousands of cells at one time. We can look at the immune cells and the tumor cells at the same time. But this sort of device costs over $100,000, and it's really just one of many of the new devices that Salk researchers need to go after some of these bold questions that we've all been talking about today.
Rich. I saw Rich's hand go up. I got to call on him. Just because he's on the trustees with me too. So.
When we think about metabolism, which obviously you spent a lot of time on today, one of the biggest breakthroughs, when you think about it from a human health perspective, has been the link between lipids, right? HDL and LDL metabolism. And you can identify people at high risk for cardiovascular disease very, very early. And then obviously the development of drugs like statins, which can intervene in lowering LDL cholesterol. So one thing that you didn't talk about today is, one, are there approaches that either you or others are taking to identify cancer early? And ultimately, will there be a simple blood test, for example, just like we have with LDL and HDL cholesterol? And then will that be the stepping stone and foundational work, like what Sachin talked about, to then say, okay, now we can develop new therapeutics in those areas?
Yeah. Yeah. Okay.
That's a great question, Rich. And, this path to biomarkers, predictive biomarkers, begin with basic science research, starting maybe with laboratory experiments and then small clinical trials and so on. So at least in my labs, in our team, we have some understanding. So for example, we know that our gut microbiome interacts with food and digestion, that process, sometimes to produce some metabolites that may be carcinogenic. And that might be the reason why people who work day and night, they're more prone to colon cancer. And what we found is by this optimizing circadian rhythm, we reduce the level of one metabolite, TMAO, which is also linked to cardiovascular disease and colon cancer. But this is a small trial. So now we're expanding it to 150 people. And then if we find the same signal is there, we may expand to people who are at high risk for cancer, for example, firefighters and night shift workers, and then see whether that is a predictive biomarker of reducing or predicting the cancer risk. That's just one example. Thank you.
I add on to that. I mean, the question originally was, are we looking for biomarkers to find cancer early? But I think beyond that, we also need to be looking for biomarkers that predict response early, right? So then we can know right when we see their cancer, oh, this is what's going to work for you, or this immunotherapy is going to work for you, or this autophagy inhibition is going to work for you. Because the reality is sometimes we're wasting 2 years of treatment time trying to find that right treatment. And so understanding what these tumors look like right when we see them at first biopsy, and being able, again, to peer in and understand how these networks are working together, that will allow us to create their first round of therapy to be their best round of therapy.
Yeah. One other thing, Rich, that we're doing, there's a consortium of some of us that some of you may have heard of before. It's a couple of years old now, but when I first took over as Cancer Center Director, we signed on a collaborative agreement with a huge kind of multinational conglomerate corporation that is effectively embedded with and part of hospital systems in Europe, but now also in the States. So like now they're merged with Northwell and some things like along the Northeast. This just happens to be the largest tumor biobanking operation on the planet. And they, in addition to collecting vast, larger amounts due to different regulations in Europe of normal tissue, normal adjacent with tumor samples, they also have been collecting blood samples at every intervention point. And we were able to obtain from them 2 years ago now, and for 2 years we are slowly, with Christian, with Alan, and with Sue, we have started going through this massive inventory using mass spec to focus in on mostly lipids first, but to look for unique biomarkers within the tumors. We haven't even gotten to the plasma yet, but we have the plasma. So that would be an excellent thing to look at. And then, you know, my hope or a dream that we'll layer into that is also for certain types of cancer, like colon cancer and lung cancer, we have good genetic mouse models that have different genetic variants that match the human counterpoints, so we can look for conserved lipid signatures or conserved even other metabolic changes, lactate changes, pyruvate changes, you know, creatine in the blood. And so that is on the books. That's like a very big initiative. We have a little bit of funding for it now, but that is one of the things that actually will fall under this umbrella of this team. So.
Great. Okay, we're going to wrap it up by since you are the director of the Cancer Center. I'm going to keep you on the hot seat. One final question. What gives you hope for the future of cancer research? I think it's a good one to end on.
Yep. Yeah. I think. I mean, I've been working on cancer, for the past 30 years. I'm 1 year. I'm the same age, basically, as the Salk Cancer Center. A few things were founded within the same time period. And never in my lifetime of working on cancer, I mean, we've seen in the past 10 to 15 years, the first results and the first fruits of targeted therapeutics and using personalized medicine. There are people on our board who have been treated continuously and personally represent the outlier points of the longest anyone has ever lived with a type of cancer that used to universally kill people within 2 years. Immunotherapy is providing cures for, again, a sliver of patients. I think we have every reason to believe that with the advent of new technologies and with combining people across these disciplines, we will develop new diagnostics and we will come up with new therapeutics that represent a third wave of cancer treatments in the next 5 to 10 years at the most. And it's a really exciting time to be a part of this history of developing new drugs. I mean, put simply, the hope, unambiguously, is that the next generation, right, like our kids, my kids, like my grandkids, that cancer for them is going to be a manageable disease. It is going to be a disease that people get, and you get a medicine for it, and then you move on, the way many other diseases are treated. But this is not going to be the death sentence that it has been for all of human history. So that's what we are committed to trying to make happen. That's our deal. So thank you for coming tonight.
That's great. Well, I think what gives us hope, and those that are on the trustees and those involved with Salk, is that adding the skill set and the team, and this is, well, we skipped 1 year because of COVID, but this would be the 4th Conquering Cancer Summit. So to just see the momentum and the researchers and you guys, the momentum and the basic research that you guys are bringing is great. When President Biden kicked it off here 4 years ago, it's just amazing to see the progress we've had since then. So it'll be really exciting when we're here 4 years from now. But before we wrap up tonight, we're excited to announce, keeping with the times, a special crowdfunding launch. As you heard tonight, technology is critical to cancer research. I'm going to look at my notes so I don't screw anything up. But tonight we're launching a crowdfunding campaign to raise $100,000. So we'll have a call to action. It's too bad the presentation auditorium isn't full tonight, but the good news is we've got Zoom, so we're reaching out to a lot more people. A campaign to raise $100,000 to fund a new state-of-the-art live cell imaging microscope for Salk's Cancer Center. Specifically, the microscope captures high-resolution images rapidly and in real time, allows scientists to better understand how tumors disrupt cellular processes, which we heard a lot about tonight. Technology like this is critical to drive collaboration and innovation discoveries here at Salk. This is a tangible way in which each of us can all help. As we provide the newest and latest technologies for the scientists working so hard at the cures that mean so much to all of us, we hope that you will consider a gift to our Cancer Center crowdfunding program. Thank you. This concludes our program. Thank you for the participants via Zoom, and also those that are here in the auditorium. Thank you for your support, and let's hope we get more momentum here in 2021 and 2022. Thank you.